Hi, and welcome to Q-rious. I'm your host, Nathan, and today we've got two experts on to talk about two different types of targeted sequencing. So, let's get curious. Targeted sequencing revolutionized genomics, and today we're going to be talking about the methods and considerations for amplicon-based and hybrid capture-based targeted sequencing. Remember, you can always ask questions in the box to the right hand of your screen. Joining me are Ellen and Marcus. Hi, how are you?
Hi, very good. Thanks for being here today.
Nice to be here.
So perhaps you can get us started on what exactly is targeted sequencing?
So targeted sequencing is all about focusing on certain region of the genome or transcriptome. And because you are selecting a few targets, you can actually get higher sequencing depth. And higher sequencing depth means more accurate and reliable data. And also, if you think about it, if you focus on such a small subset of region of the genome, it's way more cost effective. So it's also better to have more sensitivity to be able to respond to a particular biological question or clinical case.
So you can really zero in on specific regions of the genome, right? Like genes or biomarkers or antimicrobial resistance genes.
Exactly, exactly.
Okay, great. Marcus, can you tell us what are the different types of targeted sequencing?
Yeah, for all targeted sequencing, we need to keep in mind we first always have to select these molecules that we want on the sequencer in the end. And in the lab, we call this process of the workflow the target enrichment. So there are many methods, but the most commonly used ones are hybridization capture and amplicon-based sequencing. Yeah.
So how does hybrid capture work?
Hybrid capture is quite a long workflow. So you start up with whole genome libraries that you need before starting the hybrid capture. And these library fragments, they represent the whole genome at the moment, but we need to select the molecules that we want. And to pick those, we use double-stranded DNA probes, which carry a biotin, and they are DNA fragments themselves, like our library. Mm-hmm. And they hybridize, and they're magnetic, because we use streptavidin beads. We pull down the probe with the biotin and the library fragment and wash the rest away. And this is how we get our library.
And then you just sequence those parts.
Exactly, the rest is gone this time.
And amplicon-based?
Amplicon-based sequencing is a bit faster. You don't need this whole genome library prep before. You just start off with your genomic DNA, for example. There's a fragmentation to cut it into smaller pieces. And the libraries are simply generated by ligating platform-specific adapters to it. And then comes the enrichment. And for that, we use primers, like in a PCR, really. And these primers only bind to our target molecules. So PCR is only going to take place on our target molecules. And this we do in two series of PCRs. We add some index primers to that to get our ready-to-sequence libraries. Well, because these adapters that we ligate, they carry unique molecular indexes, that makes this method ideal for oncology applications, as we can trace back every single input molecule and can be highly sensitive with these panels.
So I think that we do have a short video about that. Ellen, can you tell us more?
Yeah, so we're going to watch a video featuring Dr. Barnaby Clark. He's working at King's College Hospital in London, and he's going to tell us about how he uses targeted sequencing in his lab.
Okay, great. Let's take a look.
Hi, I'm Barnaby Clark, clinical scientist based at King's College Hospital. I'm the laboratory lead for precision medicine and responsible for delivery of genomic testing. We've got gene panels in service delivering myeloid disorders, lymphoid disorders, adult neurological brain tumours and pediatric tumours gene panels as well. So we've designed a very small 100 amplicon human identification panel in collaboration with QIAGEN. We're using QIAseq panels for MRD monitoring by sequencing. We then, using the diagnostic gene panel data, we can identify mutations that we want to use as markers to monitor these patients going longer term. We are restricting the region of interest down specifically to the actual patient and the patient's tumor profile that they had, and then we're sequencing a smaller region of interest per patient so we can maximize the amount of sequencing depth and reduce the cost. So we're hoping to be able to sequence on a MiSeq sequencer 8 to 10 individual patients with redepth up to something like 20,000, hoping to get to below 1% allele frequency detection. This is still a research project, but at the moment the data is looking promising and we're quite hopeful. We're looking at Q-rious targeted prochemistry and its application for MRD monitoring using sequencing, and also to see if we can develop a non-invasive prenatal test for sickle cell disease. Firstly, the hands-on time of the library prep is down to around 6 hours, which means that we can do a library prep for a gene panel within a day, and then the next day we can quite comfortably kind of normalize and load onto the sequencer, and potentially with an extended day we could potentially deliver this in a whole day. So that's very attractive for cancer labs where turnaround times are short. The other part of the chemistry that we like is the minimal number of bead cleanups that are required in the library prep. Instead of having bead cleanups, it's enzymatically treated at every stage in the library prep, which means that the number of starting molecules within there stays the same. So your complexity of your library should, in theory, be better. And this is important in applications where you've got very few template starting molecules, such as in MRD monitoring.
So we just saw how they use Q-rious amplicon-based technology at King's College. Dr. Clark mentioned that they get the best of both worlds. What did he mean by that, Marcus?
Yeah, indeed. I think he referred to what our enrichment technology does. So this Q-rious enrichment technology is what we mean for our amplicon-based panels. And so they are fast, so you like the speed of the workflow a lot, the accuracy of it, and it's perfectly suited for these smaller kinds of panels that he's using. So, and the actual special thing about it is how we enrich our targets. So we have only one single primer that is binding to our region of interest. For a PCR, you need two primers. So the other primer is a universal primer. And by combining these two, target-specific and universal, we can make our target enrichment very uniform, and we can also make it very specific. And this is reflected by a very equal and even coverage distribution throughout our target region. The other big strength of these panels is the input, actually. You can start with quite low input amounts, and they're ideal to use difficult input material, like material from FFPE sections or from liquid biopsies, such as from plasma, from urine, circulating cell-free DNA. And therefore, the whole method is ideal whenever you have to deal with somatic mutations, because this gives you this extra boost of sensitivity when you have the need to call variants with super low frequency.
Yeah, I think that's really important in a lot of different oncology samples. But what about range? Can you use it for anything? Are there any limitations?
The Q-rious solution can handle a vast variety of sample types and analytes. So in terms of analytes, obviously, you'd need to extract DNA or RNA from your sample. And actually, you could even run both nucleic acids in a single tube workflow. In terms of sample types, as Marcus just mentioned, the Q-rious technology can be suited for all kinds, like obviously high quality nucleic acids coming from cells, frozen tissue, but also challenging samples like FFPE or liquid biopsy. So in liquid biopsy, you would extract circulating tumor DNA, and you would need a really high sensitivity to analyze that type of sample. And the Q-rious technology is very well suited for that.
And the best thing about it is actually you do not have to stick to our catalog panels. So it's fully customizable, actually. You can just go to GeneGlobe, pick your genomic regions, and enter it to this user-friendly interface that we got there. and have the algorithm do all the work for you to design and optimize your panels. And if you don't like that, well, you can just get in contact with our experts at Enterprise Genomics Solutions, and they will design and optimize the panel for you. Or once you have your NGS results, you get in contact again and design validation assays with qPCR or dPCR, for example.
Yeah.
So Dr. Clark also mentioned turnaround time in the video. How important is this for high throughput labs?
Yeah, actually, the classic protocols are very well suited to be used with liquid handler or NGS instruments. So basically, because you're going to use an instrument, you can go for really high throughput, as you just mentioned, but you can also increase the reproducibility. So less hands-on time for your lab technicians and higher reproducible results. So it's a seamless integration of the solution into a lab to actually go for a high number of samples being dealt with daily.
Okay. So we haven't talked much about hybrid capture yet. Marcus, can you tell us what are some of the advantages and differences of hybrid capture?
Yeah, well, for hybrid capture, you can target total different range of target region size, let's say. So the targeted panels are rather for small panels, let's say 500 genes tops or maybe less. You could do hybrid capture with that, but to do targeted panels is just more suitable. The workflow is easier. You can make use of this increased sensitivity that you have there. But when you have larger panels with hundreds of genes, like our CGP panel, or thousands of genes, like a whole exome, for example, then hybrid capture is definitely the way to go. You can capture material from DNA, from RNA, and with that you generate a whole wealth of information. You can assess like biomarkers and signatures like tumor mutational burden, microsatellite instability, homologous recombination deficiency, etc., etc.
Yeah, I think we have a video where the researchers really needed this high complexity and uniformity. So why don't we take a look at that?
Yeah.
Hi, I am Dr. Oğuzhan Bahadır. I am the head of Diagnostic Laboratory. And Diagnostic Laboratory started to serve as a tissue typing laboratory in 2017. This project is still ongoing, and more than 1 million samples registered in the bone marrow donor bank of Turkey have been tested so far. There are currently two main approaches to analyzing targeted NGS threats: hybrid capture and amplicon-based. Although both methods have their advantages, factors such as no allele dropout, more uniform data, sequencing of complex genomic regions, and less amplification bias favor hybrid capture over amplicon-based. Therefore, we use this method extensively in our laboratory. Quality is not a simple concept that can be assessed by looking at only one parameter. The quality of a result should be assessed by evaluating different parameters together. Median coverage, average coverage, on-target rates, and coverage rates with thresholds are important parameters. Of course, uniformity is an important quality parameter because of its impact on CMV analysis and because it shows that the data is more consistent among different samples in same batch, also other batches. With the QIAsec Human Exome Kit, we experienced improvements in many quality parameters, including the ones I mentioned before, like median coverage, average coverage, and of course uniformity was one of them.
So in the first video, we saw amplicon-based sequencing. But here it was a bit more complex, so they chose hybrid capture instead. Marcus, can you comment on that?
Yeah, so what Dr. Badish explained, they did a really impressive number of whole exomes there. So, and whole exomes, they are a very large target region. We're talking here about 30 megabases of size or more. And so he was absolutely correct to pick hybridization capture as the best way of enriching that. And actually with hybrid capture, you can not only go for DNA enrichment, you can also enrich from RNA libraries. For example, our new CGP panel for comprehensive genomic profiling. It uses multimodal libraries from DNA and from RNA to enrich 724 genes on DNA level, but also an additional 200 genes from RNA level. And from that you get, all in one assay, you get such a ton of information that is extremely useful for translational research and precision medicine.
Yeah, I agree with you, Marcus. And hybrid capture is also a very flexible technology because it can enrich so massive amount of region of the genome. You can call for single nucleotide variants, but also copy number variation. You can assess some genomic signature like tumor mutational burden, microsatellite instability, homologous recombination deficiency, and on the RNA side, you can also call for gene fusion and exon skipping event. So basically, with that kind of technology, you get a full picture of the genomic and transcriptomic landscape of a single sample. And because you can multiplex many samples together, it's very, very efficient and precision medicine, as Marcus was saying.
Okay, but how is the uniformity across regions?
Yeah, uniformity is probably one of the biggest strengths of our X-Hype product lines. So this was also mentioned in the video, that the uniformity was quite strong and was probably the reason why the QIAseq exome and the X-Hype human portfolio was chosen there. So the uniformity means that we have a very balanced coverage across all target regions. And in hybrid capture, this can be difficult sometimes because there are regions that have low complexity, or they are extremely rich in GC content, and this makes capture difficult. But we have our double-stranded DNA probes, so they allow to capture both strands of the library, which helps the situation. And this, combined with our chemistry, can balance out the uniformity fairly well, actually. And this allows to sequence less average coverage. You have to resequence less, and by this you save lots of sequencing cost, actually.
So Ellen, what's a quick and dirty summary, the pros and cons of amplicon-based methods and the hybrid capture-based method?
Okay, so let's start with amplicon. As we were explaining, it's great if you have a low sequencing budget and you want to focus on a smaller subset of the genome. It's going to be quick, it's going to be high throughput, and it's going to save you money with that lower sequencing budget. However, it might actually miss out if your target mutates rapidly. You might need to go through several iterations of the design, and you might miss out on some very complex region. On the other hand, hybrid capture is indeed a bit of a longer workflow, more hands-on times as well. But if you want to screen through really vast region of the genome and transcriptome, that's going to be the best technology for that. It also can call structural variant very, very well. So these are a bit complementary to each other, and depending on the case, you can choose one or another technology.
So they're letting me know we have some questions from the audience. Do you guys have time to stick around?
Yeah, of course.
Sure.
All right, great. So the first one is, how does the amplicon-based method compare with hybrid capture in terms of sequencing depth and variant detection sensitivity?
Okay, so with an amplicon-based technology such as the QIAseq enrichment technology, you can actually go for a very deep sequencing of a fewer number of targets. That makes it ideal for, let's say, calling very low frequency mutation, and this applies, for example, in cancer. Hybrid capture, on the other hand, you're going to go for lower sequencing of larger regions. So it's great for the structural variants. It's great to have a full picture, comprehensive profiling of your sample. And actually, it's also great for fragmentomics. So yeah, that's what you can get from both technologies.
Okay, great. So the next question is, how much DNA do you need for amplicon-based sequencing? And what about hybrid capture?
Yeah, they're not too different, actually. So for amplicon-based sequencing, you can go with fairly small amounts, like 10 nanograms of DNA, for example, that should do it. Also FFPE samples, doesn't have to be that much. It's always good if it can be more, but many users, clinicians that work with these difficult materials, they don't have so much of this precious material, so 10 nanogram will do. For hybrid capture, you could also start with 10 nanogram, but we recommend to go for 20 or for a solid 50 nanograms. This will really deliver highly complex libraries.
Mm-hmm. So more complexity, just a little bit more DNA is recommended.
Yes.
Okay, great. The last question for today. Are there any specific advantages of the QIAGEN hybrid capture panel versus other companies?
Yeah, I have to come back to the uniformity that we have talked about earlier. So our double-stranded DNA probes and the chemistry that make this great uniformity and help you save sequencing cost is certainly something that is pretty outstanding of the QIAseq panels. For the other points that stand out pretty much, I would like to focus on the workflow itself probably. So the first thing would be the speed. We can run our hybridization in a very short time, actually in 30 minutes only, which is very, very fast. We can run it longer, 4 hours overnight, but half an hour will do. And many competitive products out there, they forcefully need this. They need the 4 hours or overnight or even 2 consecutive hybridization reactions. So we have one, 30 minutes will do the trick, which saves a lot of time. And then maybe the capability of automate our product is quite something. It's not so difficult to do, so you can pick a liquid handler if you have a high throughput laboratory, have the machine do the work for you. And last thing to mention would probably be the ability to customize. So like we talked earlier with the gene globe, we can do this for our amplicon-based panels, but we can also do this for hybrid capture. We can design fully tailor-made probe pools to enrich the target region, very individualized.
Okay, so they're customizable.
Yeah.
They have a shorter time, 30 minutes sounds a lot better than 2 days.
That's very fast.
And they're highly uniform. That's sounds like a lot of advantages.
Yeah.
I think that's all we have time for today. Thank you both for being here.
Thank you, Nathan, for having us.
Thank you.
It was great.
And thank you too for joining us. I know I learned a lot today. I hope you did too. And join us next time. Until then, stay curious.